Good evening, Dr. Shobha.
Good evening, Bhavani.
Yeah. Shall we begin?
Yes, Bhavani.
Okay.
We wait for another 3 minutes.
Yes, no, not an issue.
Yeah.
Dr. Manjusha, do you have any questions? I could see you have unmuted yourself. Do you have any questions? Well, I assume it's by mistake. Dr. Shobha, let's start this.
Yeah. Okay. Good evening, participants. So today we'll be seeing about Dr. Duke's ethnopharmacological database, followed by chemical structure drawing tool. Any queries in the previous session? So you can ask question. Otherwise, I'll be going into a session. If there is no question, just give a thumbs up symbol. We'll proceed with the session. Any queries? If there is no query, just give a thumbs up symbol. So I'll be starting with my presentation.
Well, I could see few thumbs ups.
Okay. I just open just a minute. I'll start with my presentations. Today we are going to see about Dr. Duke's ethnopharmacological database, that is phytochemical compound database. And followed by, actually, I have taught you already how to retrieve the smiles and SDF format from the PubChem. But for this chemical structure, which was not available in the database, we need to go for ChemSketch. And synthetic compounds or modifications can be done with the help of ChemSketch software. And optimization also have to be done over here. So we have to use ChemSketch for preparation of compounds. So first, let me give a brief introduction about Dr. Duke's phytochemical and ethnobotanical database. So it was started by Dr. James A. Duke, and the name of the scientist who discovered this particular database. It was hosted by USDA, and it was widely used database to exploring the plant. We have many number of plants reported over here world over. All the plant data was reported over here. As we saw in the impact database, we have similarly all the activity reported, but here there was certain filter option also available, and we can search the plant by name, chemical name, with the help of biological activity or ethnobotanical use. Based on its Ayurvedic medicine use, you can get, and you can download here in the Excel format. That was a additional feature over here. And so application again, so when you know the compound data and its activity, we can definitely go for it. We can get the bioactive phytochemical constituents from the plants, and analyze its therapeutic role. So, and for our research purpose only, we are going to use that. And for more detailing activity about the pharmacological activity or mapping of the compound. So for all those things, we'll be using the compound data. So for that, that would be helpful. And supporting drug discovery, toxicological parameter, we can analyze, and network pharmacology or drug repurposing approach, and you can go in depth about the dietary supplement based upon the formulation, all those things, whether you can use the component in the fortified food. So all these things could be analyzed based upon the compound data or the data for the compound. So for that, and some quantitative data was also available here in the Dr. Duke's ethnopharmacological database. Here you have, by search, you have different names. First by plant, by chemical name, by biological activity, and ethnopharmacological activity. By plant name, you can get the list of chemicals, and you have an option to remove the ubiquitous chemical, that is common chemicals that can be removed, because and based upon the activity also, you can segregate the list over there. And by the chemical name, so if you want any specific chemical compound name alone, and you want to find that whether that particular compound was present in how many plants, that could also be analyzed. And by biological activity, find all the chemicals and plants associated with the activity. So for example, if you're going to work with anti-inflammatory activity or anti-cancer activity, so based on the activity also, you can find the chemicals and the plants related to it. And based upon its traditional usage, also you can find with the help of keywords. And based upon the ethnobotany, that is plant, specifically about plant, and based upon syndrome, disease based also, you can get. So that these are the different symbols to find out the terms. So for example, in case if you're going to search by plant name, so you can use the plant common name, or um, or the scientific name of the plant. So the result would be the list of chemicals, and for with the activity, and the general uses of the plant will also be mentioned, whether it was a traditional Chinese plants or Indian medicine or digestive problem. So whatever it was reported, all the activities, uses, everything for the particular plant would be listed, and the same data can be downloaded in the Excel format. And when we search by activity, so for example, if I'm going to search with anti-diabetic, so it will list out all the chemicals that was already reported for the anti-diabetic activity. So this specifically works. So when a people want to work with any particular disease and they want to go for kind of screening kind of thing, so then they want to work with large number of compounds or one compound from different plants. So maybe activity based, you can go with this option. Or when you want one compound with multiple plant, you can go with the compound based search, and you can find the plant which containing them. That information will also be available. So you can search in this option also here. And similarly, when we go for a chemical database, from chemical based search, so you can see that this berberine, so what were the activity was reported for this particular compound. So if I'm going to work with one particular compound, I can go and check what were all the activity reported for this particular one compound. And also, so as we know, this particular compound, we check the paper, berberine, anti-diabetic and the hypocholesteromic activity, anti-diabetic activity was reported. And in addition to that, it has antimicrobial, anti-tumor was also reported in some papers. So you can check what were all the mechanism was reported for the particular compound. And this gives one to many mapping, and from one compound to many plants. So based upon the same compound might be present, reported in many common plants. Maybe one source might not be available with us, but maybe another source, maybe we have nearby us. So that source also we can take by finding the compound-based search. And used ethnobotanical use, for example, some plant might be used for diabetes, some plant might be used for cancer. So in that case, I can use a keyword diabetes or a cancer-based disease name. I can use and search. So that was the ethnobotanical use. So where I get the plant which was reported for particular disease. So here, with the help of disease, I'm going to find out the plant. And from the plant, again, I'm going to, so this could help in polyherbal formulation kind of study. Here you can get all the data relevant to the plant and its plant-based chemistry and its phytochemical based activity could be analyzed. So here the keyword used here is diabetes. So I'll just overall coming to coming to an overall conclusion on the search page. Switch the search mode to the plant. So I have to choose any one option, plant or compound or ethnobotany use. Any one option I have to choose. So if I'm going to choose plant, I have to give the scientific name or a common name, click on to search, and the result will contain list of chemicals and its bioactivity, concentration data. If available, it has, otherwise it won't has. And references, that data will be available here. And also, from there, you can check the chemical name to see in which other plants it is found. So if you want to find, if you find one compound as a novel compound, that also you can check it. You can check it whether that particular, if you're interested in one particular compound from any one plant. So if you want to check that whether it was available in all the plant, that also you can check in the activity. And you can also export the data in the form of CSV file or else PDF file. You can find the data you can download the data in the particular format. And this is the home page. So I have given the link over here. I'll share the link when I do the hands-on session. And I'll share the PPT also. Here, you can see the options over here. This is the homepage of Dr. Duke's ethnopharmacological database. You can see A is the biological activity, C is the chemical, and E is the ethnobotany plant, P is plant, S is syndrome, U is ethnobotany activity. So you can see here, P is here, it was mentioned as list of plant, and P is nothing but plant, and in alphabetical order, it would be listed. In bracket, the family name of the plant was present. And you can see in the left-hand side, search by entity type. You have all the filtered data available. That is based upon biological activity, chemical, plant, and syndrome, and ethnobotany specifically activity and the plant. So all the data were available. So first here, just choose the plant option and click on to drop down and click on to the search option. And here you can put the plant name and click to the search option. It will list out like this. Can you see here? Like just put the plant and type the plant in the search term, and your plant name will be listed over here. And so once you click on to this particular page, it will list out to the page like this, where you can see the common name and how this plant is used. So it can be used as a food, and you can see here, you can see the chemical with activities. Exclude ubiquitous chemical. Exclude ubiquitous in the sense, the the common chemicals, acetone, kind of ethanol, these kind of chemicals, they'll come under ubiquitous chemicals. So you can click on to this ubiquitous chemical list and check what were the ubiquitous chemical. So if you require that for research, you can go use the option without excluding, you can use. Or if you want specifically, if you don't want those kind of components, you can remove the ubiquitous chemical, and you can choose the part of the plant where you're going to refine the data. So you know very well the different parts of the plant can be used for research, that is root, stem, that in plant that would be around 30 to 40 parts can be used for our research. So that data alone could be segregated over here, and those data up, were uploaded specifically for each plant, and each source data uh, were uploaded over here. So you can apply all the filters and click on to apply filter. You get the data. I have to click to the fruit data alone, and the parts per billion data also, that is parts per million is nothing but the concentration of the data of a particular component in the plant. So this data you usually found in LCMS data, reported component and the particular plant source. If they publish, they would have taken that particular data. That is, it is specifically a quantitative data of the plant. Uh, Quantitative data of particular compound in the plant. So that data, if it was available, if they found, if it is published in literature, they have taken and given over here. And you can see here in the left side, you have here it is a source, and you can see how for this, for example, this hydroxytryptamine, it was reported for 15 activity, and you can see it source of fruit, and here the concentration was not mentioned, and reference were not here. And for some, there was a reference were mentioned. So you can see the compound name, so ethylene or different components name were mentioned. So if you want specific detail, if you go and click over there, you will get the uh, data. So parts per million is nothing but a unit of concentration often used to express a dilute concentration. That is concentration of the substance. That is concentration of the particular component that present in the sample in parts per million unit. It was represented. So any doubts till now? Any doubts, participants?
Dear participants, could you please kindly give thumbs up if you're clear?
Yeah, if you have no queries, just put a thumbs up symbol. Anyhow, we are going to do the hands-on session followed by this. Okay.
Great. Please continue.
Yeah. And once I'm okay with that, I'll be downloading the Excel. So I can also check the activity also reported, and how much concentration that particular, I mean, I have, if I choose specific compound.
Dr.
So,
Sharma, sorry to cut in.
yeah,
We have, I think, yeah. Ms. Kamalika, could you please ask your question?
yes, ma'am. Yes, Kamalika. What is activity count? Activity count is, for example, anti-inflammatory, diabetic, that now how many activity reported for that particular compound. If you click on to the compound data, wait, I'll just show you. If you click on to the compound data, here it was mentioned, right? Anti-fatigue, anti-inflammatory, anti-stress, estrogenic. So these are all the activity reported for 𝐬𝐮𝐸 compound. Already reported activity in the publications. Am I clear? Ma'am, how do we ensure whether the drug is deliverable or not? So drug is deliverable or not based upon the ADMET property. We have to analyze that, and then only we can check whether the drug is deliverable or not. Apart from that, we cannot tell that based upon in this database. Definitely, we cannot find the data based upon the ADMET data. We check in Swiss ADME whether the drug was absorption was there. So those kind of parameters, ADMET parameters, toxicological parameters, that has to be analyzed. And again, the wet lab support is also necessary to study about the drug delivery process. Hope I'm clear. Sure. Hope I'm clear. Any doubts? Okay, fine. So here you can see the different activity was also mentioned. And so for activity-based Excel, if you want, you can download the Excel file, and you get the Excel data. Or plan-based data also you can activity-based data also we can search, and plant-based data also we can get. It's the same compound present in different plant. The same compound will have different activities. So all the data we can get from the search option. So first we did the search for a plant, and we click on to one compound, and that one compound has reported for many activity over here. And that one compound present in multiple plants was multiple plant parts has also been reported. So that detail will also be available in this particular database. So it will be having the complete data set of a compound as well as the plant which was reported for. So this data could be from LCMS data, GCMS data, or individual compound isolation data. So whatever data it is, it was reported. And if you want to go in depth about one particular compound or one particular plant source, definitely I would suggest you can check your Dr. Duke's database and impact database, so that you can get a collective information about one particular plant, and you can decide whether you can proceed with a particular plant or not. Am I clear? Okay. So once I complete all the information, I have to download the XLS file. So I have to click the XLS file. I'll just open and show you the XLS file. So the Excel file, it is general Excel format. Whatever table here you are seeing, you're going to download the same table, and you're going to suppose if I'm going to use these 5 compound, so it will be there. And you can take the smiles format of the compound, and you can go for your study part. Am I clear? And view the results page will contain the phytochemical that is in the table data. Whatever data were available, that is what it has shown. In table, you have first part, you have the compound, phytochemical names. And second, you have the biological activity linked to the compound, whether it is anti-diabetic, antiviral, and the quantitative column that represents the parts per million data of the plant, that is the concentration of milligram per 100 gram of the plant, and a reference scientific paper was also there. So this is the data we get from the Dr. Duke's ethnopharmacological database. So let me first complete the hands-on for ethnopharmacological, then we'll come to 𝐏𝐥𝐞𝐚. Is that okay? Or shall I complete this and go to the hands-on training part? Participant, what's your suggestion? Just tell me, or shall I complete Dr. Duke's and come back to the 𝐏𝐿 part? Yeah.
Dr. Shubha, we can complete first, Dr. Dukes, and
Okay.
then we can.
Yeah, sure. Thank you. So just open a Google and take a link. I'll just copy the link and post it in your chat. So those who are working can simultaneously work from your end. So this is the link for Dr. Duke's ethnopharmacological database. And I'll just go to the Google. Just paste the link. Just click on to enter. So here you can see the general option. You can see A is for biological activity. So based on activity, it was reported. You can see the page. So here I'm just choosing a plant and I'm going to click name. So if I put the common name, it will show the biological name also. Or if you know specific scientific name, you can click onto the scientific name and click onto the search option. So here you can see the name of the plant and family of the plant over here, common name, how it is used based on the medicinal property only. We use name. We won't take it as a food. For its medicinal activity, we use the name. So if we use a particular fruit or a kind of thing could be mentioned as food. Otherwise, in which form we use that would be mentioned. And the same plant, the chemical name were listed. You can see here that it will go on to the pages. As we know, the neem part was reported for a bark, seed, leaf, all the particular components. So you can check that also. And chemical list with activity. So here it shows around 6 pages now. So if I mention chemical list with activity and apply filter, you can see the list will come down to 3 pages. And in turn, if I mention, so you can see here the calcium, fiber content, everything was mentioned. In case if you're going to work with a nutritional kind of thing, in that case, you require these kind of basic data. So for that only, they have mentioned that particular data. So in case if you not needed that, you can exclude ubiquitous chemical part and click on to apply filters. You can see the count was reduced to 2 pages. Now, so you can see the different parts of the plant from the neem. That is bark, flower, fruit, leaf, a whole plant, seed, stem bark, and wood. So these parts were reported. Suppose if I'm going to check for a neem seed, apply filter, I can see only 8 compounds. So this is how we need to refine the compound. Or if you want a whole part, just click any and apply filter and click onto the Excel file. The whole Excel file will be downloaded. So if I'll just open and show you both the things. So if I want only the seed part and apply filter and download the Excel, that part alone will be downloaded. Okay. So once downloaded, kindly open the Excel file and check the data. So here now I have an option that all the chemical were listed and number of activity reported. So the quercetin was also present in the neem flower. So it was reported for quercetin compound was reported for around 176 activities. And so you have different compounds. So you can see totally 28 novel compounds from whole plants was reported. And if I want to specifically filter, hope everyone know the filter option in your Excel. Sorry. So here you have data. If I want to filter, you can have a filter option over here. You can see here different source were mentioned. So if I want only the seed part, so if I click that, I'll get only the seed part. So if I want only the leaf and click OK, it will show only the leaf component. Hope I'm clear. So this is how you can refine with your Excel data. Or if you want whole, just click filter again, you get the whole data. And you can see the reference data. As we mentioned specific activity, we get this kind of data. I'll just show you the raw data also without refining with activity and apply filter. I'll just download the XLS file. You can see here now. Just a minute. Any apply filter. Now you can find around 81 data, but without ubiquitous chemical. Few data might, there won't be any activity was reported, but all these data were together, but the component that was present with the plant. But the extract might have contained activity. So if you want those kind of compound to be analyzed, you can go and check for the plant extract based activity, check for the concentration whether it was reported, and that kind of data also you can take and analyze the data. So hope I'm clear till the download part. If there is any doubt, so if you are working participants, you can raise your query. So if there is any difficulty in handling this, so it is just in database search option. You're just going to click few options and apply filter, and you're going to get the results. Any doubts till now? If there is no doubt, just give a thumbs up symbol so that we can proceed. There is, hope there is no doubt. So we'll go with, so first I have checked the chemicals. So if I want to check for activity, so what were all the activities this particular plant was reported? So that was mentioned here. So here you can see that neem plant was reported for many number of activity as a whole. And specifically, if you want to check for the chemical count, that was also available. So you can see camphorol and quercetin was reported for this particular inhibitor activity. And you can see this 4 chemical was reported for alpha reductase inhibitor activity. So that information was available in the activity tab. And in some cases, there would be reference were also mentioned, and so for that also you can check the number of chemicals. Suppose if I'm going to work with any one particular activity, suppose I'm going to work with any painkiller, analgesic activity. So in that case, I can choose that particular component, and that alone I have to take and work for, and I can check for the mechanism. So based on the activity, so here it was mentioned as in the alphabetical order. So you can go and check all the tabs and each activity. So neem, it is very common plant. It has many number of activity was reported. If you take some rare plants, then the results would vary. So that is how we have to check the activity as well as the plant. And based upon the compound, again, suppose if I'm going to work with epinembin, so I want to check whether this compound has reported for how many activities. So it was not reported for any activity. That was also mentioned. And epinembin, it was present only in Azadirachta indica. It was not mentioned in any other plant. So if I'm going to check with some common source, wait, I'll just show you any common compound. So I'm just excluding ubiquitous with activity. I'm just filtering. Suppose if I'm going to check for camphorol. So camphorol, it is a well-known flavonoid. So it was reported for many number of activity. And if I'm going to check the plant for the camphorol, so all the plant that has reported for a compound camphorol and from which source it was reported, so that details was listed over here. And if reference were there, that is also listed over here. Hope I'm clear. So from here, we are just going to take the compound data as well as the name of the compound, and we are going to prepare our data in this format. So this is our first step in the network pharmacology, where we are going to, suppose if I'm going to work with one particular plant and one particular source, I'm just going to list out all the chemical name from the source of the plant. Maybe it could be from IMPACT, it could be from Dr. Duke's, or it could be from the literature point. So literature also, you can go and search for GCMG. Apart from the compound listed over here, if you find any compound from the literature, that could also be taken into an account. But why I'm suggesting database, it was actually they have took from the literature only, they have feed the data. But in case if the recent data, somebody published the recent new compound or any data, so that could be taken from the literature if needed. Otherwise, we can take from any one of any if there is a common compound reported in two database, then you can take that particular component and take the SMILES format of the particular component. So this is our first step. And for certain database, you require SDF format of the database. So SDF format of the structure. So that also need to be downloaded and kept in a particular folder. So for network pharmacology, the main thing is you need to create a folder and name the folder as I'm naming. The first file we are going to save here is compound SMILES. So whatever compound I'm going to take, I'm just going to give the compound name in the first column and SMILES format in the second column. In the SMILE format, again, we have checked with the PubChem database where you can go and search the compound and get the SMILES format and copy paste in your Excel data. Am I clear?
I have a question in chat. Could you please check with that?
Yeah. Hello, ma'am. Suppose I can't understand how to SMILES work. I'll just tell you the SMILES, how to download the SMILES. I have already told in the PubChem class. I'll just repeat again. And suppose I want to download the compounds which I have antiviral activity. How to download the compound? Definitely, I'll tell you. Here, we have search option. You have biological activity. Antiviral. You have here the activity, antiviral activity. So it will list out all the compound relevant to the viral activity. And here, so if you can see the pages around 9, almost you have many compounds reported here. And you can download the Excel file. So if you want to check all the compounds simultaneously, you can check the Excel file. You can see here the EC50 value, that is enzyme concentration 50 value was mentioned, or inhibitory concentration values for certain component were also listed. And you can see here around 344 compound listed for the viral activity from different plant sources. Hope I'm clear. Is it okay, ma? Shivangi. Is it okay? Yeah, fine. Any other queries, participants? Any specific refining process you want to know? Or I'm clear? Just give a thumbs up symbol. Any queries? No? Okay. So SMILES format, I'll just show you how to retrieve the files. SMILES. Suppose if I'm going to choose this as aloe So I'm just copying the compound name. Go to PubChem. This already just paste the aloe here. Just click on to this and so just click over here, aloe in the page. Just scroll down 2.1.4, the SMILES option will be available. You can copy paste the files, SMILES format in the Excel file. Am I clear? Okay, fine. So any doubt in Dr. Duke? So if you're working, I'll be waiting for 5 minutes. Otherwise, if you're simultaneously, if you're working, let me give the time to work. Participants, if you are working, please let me know. I'll be waiting for 5 minutes. Otherwise, If you're going to practice later, please let me know. I'll just go with the chem sketch.
Dear friends, those who are doing hands-on alongside, can you please raise your hand?
The Kamalika is doing. Okay, let me wait.
Others?
Others?
Anybody else? Well, I guess Kamalika is the one. Not an issue. Others kindly do it once you
I think
review.
one more has
Lakshmi,
raised that. Okay, fine.
Lakshmi.
Okay, so let me wait for Kamalika. If you're done, so if you're done, just put a thumbs up symbol so that I'll do with chem sketch. In some time, Pumpkin is showing error. In that case, where to get smiles? It was showing error. What kind of error it was showing? Let me look and I know. To use the filter in the Excel sheet. Okay, I'll just tell you that. No issues. So PubChem, usually it won't show. If the compound was available, it will show. Otherwise, just in the Google, if there is any hyphen or any when we copy paste or sometimes the number might vary. So in that case, Google just put aloe and PubChem. You may get the hit over here also. From here also, you can go inside to the page. In Google search itself, you can use in that case, because sometimes when we type, there would be some numbers would be there in certain compounds. So definitely it won't show. If there is a compound, big name, IUPAC name were there, because in GCMS, we get it as an IUPAC name. So in that case, when we copy paste, there would be some space would be there, comma would be there. So sometimes it might show error. So in that case, you can put over here. If you get the compound, you can go to the page directly. Okay. Last week it shows. Okay. So in that case, maybe the compound name you have to check. So refining in the Excel, suppose if I want to refine this particular column based upon the activity, I'm going to refine. So generally for alphabetical search data, A to Z, if you click, it will simultaneously arrange the compound in alphabetical order. First, the numbers will come, later alphabetical order, it will change. But if you want to refine your data based upon the plant part. So just this column, if I need, just keep the tab over here, click on to filter option, and it will show the arrow for all the tabs. But if you want this part to be checked, just click onto the small arrow over here. Hope everyone is visible with the arrow mark. So if you click on to this arrow, it will show the different option over here. Select all, bar, flower, fruit. So I'm just deselecting all and specifically selecting the which part I require. Just click OK alone. So it will list out the compound that present in the particular seed alone. If I'm clear. Yeah, fine. Just check with the PubChem error and let me know whether you're getting the same error now also. Because last week there was a server issue in PubChem, but now it was rectified. Okay. Fine. Any queries? Ma'am, which smile should we use? Canonical smiles you can use. Which compound you are just using? Just send me the compound name in chat box. I'll check and tell you. Can you send me the compound name? Or else if you have the compound structure, so now I'm going to teach the chem sketch. So you can draw and retrieve the smiles from that also. I'll be teaching you the drawing tool right now. So you can draw, cross-check with molecular formula and molecular weight, and you can use that structure also. Because in literature, definitely, if some compound, maybe patented compound, might not be available in PubChem database if the patent was filed for particular compound and they don't want to show the structure in public database, they won't publish. In that case, if they publish the compound structure in as a structure file was published in literature, you can draw that structure and you can use that. Hope I'm clear. Shall I proceed with 𝐬𝐿𝐶? If everyone has no issues, I'll be proceeding with 𝐬𝐿𝐶. I'll just give a brief introduction. It is just a drawing tool. We already installed the software, so you can such as quercetin. When I search the compound, it shows multiple compound what we pick. So quercetin, spelling E won't come first thing, and quercetin, if you want to, if you get multiple compound, just go and check with the molecular formula of quercetin, and you can also have one more option. Once again, I'll just show you in PubChem. So if I'm going to search quercetin, you can see here you have different form of quercetin. So if I put quercetin as a whole, it will list out all the quercetin, individual component also shown, and also with the combination of quercetin. You can see here, here it was mentioned only quercetin, or as you mentioned quercetin, E spelling was also there, and quertine. This is also quercetin, quertol. So all these names were also there for quercetin. So you can see here also the same molecular formula, molecular weight, molecular formula, molecular weight. So these both are the same one. And you can see here, it has quercetin 3-O-sulfuroside. So it is a combination of quercetin. It is not individually quercetin. You can see the molecular weight, molecular formula. So cross-check with that. And when you have GC-LC data, you definitely have the GCMS, LCMS data. You definitely have the molecular formula and molecular weight to cross-check. And the first one would be the best match. So that would be the ultimate your output. And this, you can see here 701 results for quercetin. That is nothing but the combination of the quercetin. Hope I'm clear. Any other doubts? Any other queries? Okay. So I'll proceed with 𝐬𝐿𝐶. I'm just showing the few steps to go with 𝐬𝐿𝐶. Just in a PPT first I'll show, and hands-on I'll be teaching you the same. So I'll proceed with the slideshow. So 𝐬𝐿𝐶, so you have the icon in your system, this 𝐬𝐿𝐶. So this is the icon for 𝐬𝐿𝐶, or in your search box, just type 𝐬𝐿𝐶. Once your software were installed, you'll be having this icon in your system, or just type 𝐏𝐸𝐸, you get the icon. Just click onto the icon, this page will be open. It will just show some information. Just click on to escape 3 times, it will go off. Or just click OK and you get the data. Suppose if I want to generate the data from the SMILES format. Why I need to get from the SMILES format? Because so when I draw, I miss out something in the structure. So in that case, I can, if I have the SMILES format in the database, I can directly use the SMILES format to generate the compound 3-dimensional structure. So here you can see the tool option were available. So in 𝐬𝐿𝐶, you have 2 options, structure and draw option were there. In structure only, all the drawing could be done, and draw option, it is different. It was used for certain equipment. So if you want to show some reaction kind of things, you can use that. But usually, it is not required for the structure preparation part. So we won't go depth into the draw part. So we are going to focus only on the structure drawing part. And here you can see the tool icon, and inside the tool icon, you have the generate. and generate structure from SMILE. So I'm just going to click on to this option and I'll get this box. So I just need to paste my SMILE structure over here and click on to the option OK. I get the two-dimensional structure of the compound. This compound is syringic acid. So where I have took the SMILES format and just pasted it, I'll get the two-dimensional structure immediately. Okay. After I'll just show the hands-on part also just for the information. And if I want to draw the same structure, what I have to do? So for that, I have to go for the, first I have to check for the templates. In template, you have different windows. Template window, template organization, table of radical, periodic table, dictionary, all those things were available. So table of radicals where you get the basic structures like benzene ring, aldehyde group, alkyl group, all these data, chemical data would be available. And periodic table, elements data would be completely available. So as you are all aware of periodic table, so these data would be generally available. That in template window, just choose the table of radicals because as you're installing first, in your window, the option won't be shown because in my window, you can see here, it shows all the icons over here. As I'm using it regularly, it shows. First, when you open, it will be empty kind of thing. So regularly, when you start using these icons, so it will be showing. So this icon is template window and library dictionary would be there. All those things would be available once we start using the data. And so once you draw the structure, we need to cross-check here in the downstairs. You can see the molecular formula and molecular weight, which will be there in the PubChem also. That you have to again cross-check. And once you click on to this table of radicals, you get this table of radical box. You can see here the chain data NC3, that is three carbon alkyl atom, 4 carbon alkyl atom, all like that. It will amines, all these things would be there. And you can see the double bond, triple bond, phosphonium group, and nitrous oxide, all the amino acids. So basic chemical structures which were required, it would be available. And you have benzene ring with the bond, without the bond, aromatic ring without the bond, benzene with the bond. So this is how alkyl aromatic groups and all the other groups would be present. Suppose, as I know, many of the chemical structure will contain benzene ring. So as I'm going to draw this particular structure, so I'm just choosing this particular benzene ring. Just click on to that, it will automatically come to the page. And if I want to add atoms, that is, I want to expand my structure. So if I want a CH3 atom, I have to click C. And if I want to add hydroxyl group, I have to click OH. And if I want to add amino group, N. And special atoms, as we require, you can click over here. If you don't find here any atoms, you can click on to this periodic table icon over here. I think everyone can see the periodic table icon. You can choose it. And so this is to select the atoms or the component that present inside in your structure. And next important thing in the structure is bonds. So you can have, if you want to have a single bond, you can take this one. First one, you can take it for the single bond. If you want to go for drawing like a next bond, continuously carbon-carbon. So in that case, you can choose a second one. If you want to draw like fatty acid kind of derivative, in that case, you can choose the third option. And if you want down stereo and up stereo bond, you can use these two bonds. So apart from that, we won't use, these are all the specific icons which chemists can use. And so that we are not going to use. So we are going to use the bond and the chemical. So we use to draw the chemical structure and to connect the bond. So this is the thing we require. So, and you have an erase icon where if you've done, so you can see here, this is the eraser icon. If you've done any error, you want to delete that, you can select that atom with this, you can delete that. And So I'm just going to draw this, but if I want to add, suppose in this structure, first I have drawn the benzene structure. I'll just show the hands-on training for drawing this particular structure, and you can try later. So this contains 3 hydroxyl group. So I have to just click O atom over here. From here, just I need to drag the mouse. This OH atom will be added. If I want to change the bond to OH to O, I have to click this particular bond node, and it will show double bond, after which the double bond will be added to the structure. Am I clear? So as we are drawing the structure, the size of the bond, the structure, that would be slightly vary because we are drawing, right? So in that case, if I want to make it neat form, the correct size of the bond structure, all those things was required because we cannot uh, randomly draw the chemical structure, right? Because every bond has specific size. So for that, you have to click on to tools again and click clean structure. You can see the difference between these two structures. Here, the double bond size was big and its bond size were increased. So you can see here all the size were optimized, and this is the final structure. As you seen over here, the same structure were drawn, and the similar structure were drawn, and finally we so in case CH3 need to be added, from here, I have to click this carbon atom and the CH3 will be added, and I can save the structure in, this is a 2D structure. You can take it for your reference. So once you draw the structure, you can publish the structure in any publication instead of taking from Google. For that, you have to give the reference. So here you are going to use drawing tool and you're going to draw by your own. You can use that for the publication kind of purpose. Then there won't be any copyright issue for publication. And after which this has to be changed to the 3-dimensional structure, where you can see here ACD Labs 3D viewer option. And so ACD Labs 3D viewer, once you click, you get an empty screen like this. So here in the downstairs, you can see here 𝐏𝐥𝐞 copy to 𝐏𝐥𝐞 3D view and 3D optimization. These things would be available. So first I have to click on to 𝐏𝐿𝐴 and go back to this. It will come back to the 𝐏𝐿𝐴 this page option, 𝐏𝐿𝐴 page option. Then from there, again, I have to give copy to 3D option for my chemical structure to come to this 3D page. Hope I'm clear. I have to initially, directly, if I click 3D viewer, your page would be empty like this. So here you have the 𝐏𝐿𝐴 copy to 𝐏𝐿𝐴 and 3D view, 3D optimization. So I have to click on to this 𝐏𝐿𝐴. And again, I have to click on to copy to 3D. Once I move to the 𝐏𝐿𝐴, it will show the option copy to 3D page, and after which it will transform to the 3D page. So once I done with this, I have to, I can change the background color. So option for that, to change the background color, I have to go to options, colors, and so you can choose white color because it would be visible more and it would be clear for publication if you choose white background. And if you want to change the elements colors also, you can change that also. So that also could be done. All the size of the radii increase, decrease, all these things could be done. So whatever view you can want, you can change. So that everything could be done. So just work with that, and any queries, we can clear it on Wednesday session. But so once you change the background to white, it would be like this. And after which you can go for optimization. Now you can see the only O atom were there. It doesn't have any hydrogen atom in the chemical structure. So once you click on to this 3D optimization, this icon, the circle mentioned over here is the 3D optimization icon, where you can click, you can see now the hydrogen bond were added to the structure, all the part of the structure. You can see the hydrogen bond in white color. The default color of the carbon would be blue color, oxygen would be a red color, hydrogen would be white color, and nitrogen would be blue color. Apart from that, if you want to change any color, as I told you, you can go to option, change the element color. So whatever color you need, you can change and save the structure. So once you're done with all these things, you can go to File, Save As, and choose the option. So File, Save As, and you have file name and Save As type. In Save As type, just click on to the option MDL mode. If you want to save it as an image, here also you can go and save it as an image. Uh, GIF file, everything could be saved over here. And if you want default format, whatever we save for our references, MDL mode format, which could be converted in the required format in the later stage. So why we are doing optimization process? So here it doesn't change much. But in case of fatty acid derivative, if you do optimization, if it has the torsional angle in the compound, the structure will be much, it will fold much. It will be like in a folded form, and it will be little more flexible enough to bind with the target. So for that only we are doing the optimization process. Hope I'm clear. Any doubts till now?
Ma'am, I wanted to ask you a question.
Yeah, ma'am.
Ma'am, can we use chemsketch for joining or combining two molecules with the help of a covalent bond, rather?
Covalent bond,
Or
bond
combining two.
connection. Yeah, you can combine a compound, you can modify the compound if you are
No,
very
no,
specific.
I mean,
Ah.
we have compound A, we have peptide B. We have two molecules. One is the peptide, and second one is a compound chemical. So, can we, with the help of chemsketch, combine both of them together
No,
to
no, we
make them one
cannot.
compound?
No, we cannot create a bond between a compound using a drawing tool. For that, you have to do the docking procedure, where in protein part, you can give the peptide structure, and in compound part, you can give the particular compound you're mentioning, and you can do the docking. Then it will automatically create the bond between the structures. So docking is a process to find out the interaction. Suppose you're telling covalent or electrostatic or hydrophobic hydrogen, whatever interaction that form between the particular peptide and the compound, that could be checked via molecular docking approach. So docking also were there in our study. Last part is the docking. Once we find out target, we'll be doing the docking analysis. So in that, maybe in protein part, instead of protein structure, you can use the peptide structure and go for docking analysis.
Okay, ma'am. Thank you.
Okay, thank you. So with this, you can save it as MDL mode. You can give the compound name, and .mol is the extension of this file. If you're going to save it in JPEG, so you can choose JPEG option and save it as an image file. But the structure file have to be saved in mol format, that is MDL mol file. You have to save the compound structure. So I'll just show the hands-on session, so it will be more useful for you. So I'll just open ChemSketch. So like this, you have to open the file. So if you're going to work simultaneously, I'll be a bit slow. Otherwise, I'll just teach you right now. Later, if you can practice and clarify the doubts tomorrow. So first, let me draw one compound fully and explain you clearly. Then maybe next step, I'll just slow down and I'll just let you make you to do. Is that okay for all? Okay. I'll just go with the drawing part. So here you can see once I open, it shows something like this. Just click escape or just click OK. Just click escape. It will go off. And first I'm just going to tell with the open babel, like I'm just going to take a compound. Suppose if I'm going to, I'll just take a little bit complex compound so that, so I'll just take the smiles format. And in ChemSketch, tools, generate structure from smiles, paste the structure compound data. You can see here, it was pasted as per in the PubChem page. Whatever the structure file was mentioned, based on the structure, it was the structure will be pasted. You can cross-check with your molecular formula and molecular weight. Here you can see C33H40O22. Molecular weight is 788.7. So here you can see C33H40O22, molecular weight 788.66. So it was around 0.7. So this is the 2D structure. So once we're done with this, here now in the downstairs, it won't show any 3D icon. So once you click on to ACD Labs 3D viewer, it will show the empty page first, then come back to ChemSketch icon and give copy to 3D. You can see now with the help of mouse, just press and rotate. Just in the left icon, just click and just hold it and rotate. It will be rotating. Okay, and you can see the different forms. This is the line notation. This is the stick model. This is ball and stick model. Usually you can see in many literatures. This is CPK model, and this is peer model, and this is cartoon model. So you have different models. So usually you would have seen this particular model. So many literature would have seen this. And if you want to change any colors, so you can go to option, go to colors. You can change the background color to white. And just see your background will be changed. And if you want to change the atoms, so if I want carbon atom, hydrogen atom to be a different color. So usually, carbon I'll just change. If I want my carbon atom to be blue color, so dark blue color. Just if I change that, this color will be changing. The carbon atom color alone will be changing. So if I want to change oxygen atom to different color, I can change that also. And so once I done with all these things, if I'm okay with the structure or that's it, fine. And if last part would be 3D optimization. Now you can see based upon the carbon valency and oxygen valency, the bonds will be satisfied. For oxygen, there should be 2 bonds, and for carbon, 4 bonds. So that would be satisfied, and based on the double bond, the hydrogen bond will be added. So you can see once I click the optimization, the structure was little bit folding. So this folded form is nothing but the optimization. For this part only, we are usually using the ChemSketch to do the optimization. So if I do if I take the optimized structure for molecular interaction or docking analysis, that would be more useful, and to predict the interaction, the binding would be like a lock and key model. So whatever it is, so it will be go and bind with the target as a lock and key. So now you can see the structure was folded. Initially, it was very rigid. Now you can see the folded form of the structure. For this part only, we are doing the optimization, and we save the file as MDL mol format. I'm just going to file, save as. I'll be just creating a file. So I have the file over here. I'll just save the compound name. I'm just making it a square certain. Just give the short name or extension. I have to change over here. I have to choose the file type MDL mode .mol and click onto the option save. Your structure file would be saved in the MDL mol format. So this is the file I have saved right now. You can, if I open in the notepad, it will be visible like this. You can see the x-axis, y-axis, z-axis, what were the atoms over there, that details will be mentioned. And completely the structure file of the compound data. Okay. If I'm going to draw the chemical structure, what I have to do? So I'll just delete this. So if I don't want the structure, just select all, just click delete in your keyboard, it will delete, or go to file, new, click, it will come as a new file. So if I'm
Excuse me,
going
ma'am.
to, yeah, ma'am.
Ma'am, the ChemSketch version that you are using is a freeware or is it a paid one? Because
Freeware, madam.
ma'am, I
Freeware.
downloaded one software and it says ACD Chem Basic. So where from should I download the one that you are using, ma'am?
I have given the link already for ChemSketch download.
Ma'am,
Just
is
a
it the
minute,
chat?
ma'am. Yeah, I'll just share, ma'am. ACD ChemSketch, just Google and download the free chemical drawing students. Free
I'll do
chemical.
again.
Wait, just a minute, ma'am. I'll just share you the link, ma'am. You have
Yes,
to
ma'am.
register for it.
Yes, ma'am.
So here you have, right, ma'am. I'll just share the link. It will ask for some registration, ma'am, your mail ID, academic purpose like that. It will ask, just register and download, ma'am. It will be free.
Okay, ma'am. I'll try this.
This
Thank
only
you.
this detail it will ask, ma'am. Yeah.
Yes, ma'am. Yes, ma'am. Thank you.
Okay, fine. So once, so hope I'm clear till now. So we'll draw the structure for one particular compound. So this aloe itself, I'll just draw. I'll just take the structure file. So it has 3 benzene ring. So I'll just keep it like this and draw and show you. Hope both the screen are visible for everyone. So I need a benzene ring first. So with the bond, I just choose the benzene ring. And I'm just clicking 3 benzene rings. Okay. And so I want to change a bond. So the position of the bond varies. So I'm just clicking over here. It first shows 3 bond. Then the bond will be removed. So I'm just removing the bond first and adding the bond. If I'm too fast, please let me know. But I'll try to be as much as slower. So I have just adjusted the bond in the 3 data. So now you can see here the oxygen atom. So I have to click oxygen. So the default bond selection would be the first one. This bond selection have to be there. So I need oxygen atom. So I just click and drag, OH will be added. And again, I have to click over here, OH will be added. OH will be added. And OH will be added over here. So this is double bond O. So I'm just clicking the bond. It will change to double bond. Here you can see the connecting atom. So this, if there is nothing mentioned and there is some atom over there, it is CH3 atom. So you can click on to see. CH3 bond will be CH3, that is methyl, methyl group will be added. And again, I have OH bond. So OH bond will be added. So to now the structure you can see here. So I have drawn the structure. I need to cross check the structure. I didn't put the double bond over here. I'm just checking the molecular formula right now. So you can see here it was 272 and C15H12O5. So I didn't put the double bond. So the formula was varied. So if I put the double bond and everything was correct, now you can see the molecular formula 270.24 and the molecular weight 270.24 and molecular formula C15H10O. So this is how we need to draw the chemical structure. So if you want a template window, so just go to template window, table of radicals. This would be in the left side right now. And so this is, you can add and save the template for later use, but this is the template window you get. And once you're done with all those things, so you have the entire structure right now. So tools, just click on to the clean structure. So now you can see your structure was rotated, but the structure and the molecular formula, molecular weight remains same. Then your structure was right. You can keep it in any form for your reference and you can convert it to 3D option. So as I have already opened the 3D window, it shows here copy to 3D right now. And if you want to generate a smiles from the drawn structure, tools, generate. This part was not there in the PPT. Kindly note down. Here, if you want to generate for a newly drawn structure, the smiles notation, go for generate. Tools, generate. Smiles notation. So the smiles format of your data will be represented. Just click escape. So you can see the smiles format. So this data, if you click escape, the selection part will be going, so that you can copy this and put it in the WordPad or Excel. You can save it for your later reference. Am I clear? Okay, no queries. Okay, I'll go with this. So once I'm done with this, I have to copy and keep it for my later reference. So I'm just converting this to 3D and showing you once again. Just click on to copy to 3D. So as I have already chosen the color, it shows default color. Just optimize and file, save as .mdl mode, whatever folder I need. I can choose the folder and save the compound name aloe just save this aloe. But when you save the compound, just avoid using the hyphen uh, and numbers in the compound data. So that would be better, because some docking algorithms, the name issues will be happening. So that can be avoided, and large names can be avoided. Short names can be used to save the compound data. Hope I'm clear till now. So anyone, if you're working, if you find any difficulties, you can ask the queries. So this is the working part. So apart from this, if you want the stereo bond, so if this I'm just showing, in this particular it was not there, but if you want to change the stereo bond, you can use this. If you want to change the stereo bond to normal bond, you can use this and do. If you want to use benzene, fatty acid kind of derivative, you can use the third option. So all these things, once you work with the structure part, you can work with all these datas, and you can, based upon the structure data only, we can definitely work on this. So whatever data you require, you can modify and you can use. And if you want to add some charges, so mostly for docking, we won't add charges, but charges you can add. And you have the periodic table. So whatever atom you require, you can, whatever metal atoms you require, you can copy paste. But usually in Autodock, the metal docking won't be possible. But you can use any other docking program if you use the metal-based compounds. And if I want to delete this, I can use the delete option to delete the structure, or this part I want to delete, I can delete. And if I want to join, I can join. So 𝐏𝐥𝐞𝐚𝐬𝐞, first time when you draw, a little bit like the selection and attaching all these things, because when we click it, definitely if I click somewhere, it will definitely click, it will show an atom. So if you, by mistake, if you click atom and save the file, this atom will also be saved. And when you have two structure in a file, definitely that cannot be converted into 3D. So I'll just show you that also, because when you have three structure, optimization itself will you cannot do it. So that is also, it is an error kind of thing. So in that case, you can use delete option, and you can cross check with the data, and you can finally save the file in a required format. Hope I'm clear till now. Any queries, participants? Any queries? Ma'am, I'm not getting the 3D molecule. In your window, you have you checked for the 3D where option, ma? You didn't get this option? Lakshmi? That's what I told you. First time when you open now, you won't get that. So in the downstairs, you may have a 𝐏𝐥𝐞, copy to 𝐏𝐥𝐞, 3D view, and optimization option would be there for you. So like this, your option would be there. Go to 𝐏𝐥𝐞 back and copy to 3D, then give. Am I clear? That's what, Ma. Like, have you drawn the molecule in the 2D format first? Have you getting the structure in 2D format? Okay. Um, Once you give copy to 3D also, you didn't get molecule over there. Just check with, just once you go there, just click to copy to 3D and check. But yes, ma'am, but it showing in 2D. That's what here you have copy to 3D option, right? Have you clicked this option? Just follow the procedure and check. Even though later also, if you didn't get Wednesday, well, let us check with the option. Any other query? So you can switch on your mic and ask your query also, not an issue. So some of any queries participants, so ChemSketch, it is generally for any chemical structure you can draw and take for your research. For that only I have taught you ChemSketch. Now you're getting. Okay, Ma, thank you. Any other queries participants?
Jisha here. Ma'am, I have a doubt. Is it
Yeah.
possible to derive functional monomer and cross-linkers from a target? Any computational method or tool is available, or is it possible?
I'm not getting. You are asking about protein, Ma? Monomer, huh?
No. Functional monomer, and I'm talking about polymerization process, ma'am.
Carbohydrate-based derivative.
No, we have a target, and is
Okay.
it possible to derive monomer and cross-linker from that?
In target, what you're talking about protein or a carbohydrate, because each molecule is
Ma'am,
different.
target molecule.
Protein molecule, is it
Ma'am,
protein or a carbohydrate?
ma'am, anything.
If you're telling polymer means polypeptide is a protein. Polymer
Okay.
is the like, for example, plastics, bioplastic means it is a
Correct,
polymer,
ma'am. Yeah,
right?
yeah.
If you're going to tell about polymer, like you can take a branch of it and do the thing. Not the entire thing you cannot take. You can take one particular branch. It will be having the same mode of action, right? In case polymer, you can use some 3 to 4 subunit. If you are working with carbohydrate molecule, a polysaccharide. In that case, we will take glucose 3 to 4 unit of glucose. We cannot go for entire polymer because as the structure enlarges, like we break down and take the structure and you can make it you can check the mechanism. For mechanism purpose, you cannot take the entire structure, but you can draw like maximum number of units you want, you can draw and check to that your rotatable bond doesn't go beyond 40 atoms. So that is the ultimate thing. So if more the structure, then anyhow, the mode of interaction is going to be same, even it's the 3 to 4 moiety or number of moiety increases also. So that you can do, not an issue.
Okay.
But network pharmacology, I don't know, but generally mode of action you can do for any kind of molecule. Polymer, monomer, all the molecules you can do and you can draw the structure because polymer, even I have done a work with a polymer by polymer with the dye I have checked the interaction. So based on the requirement, which compound will act on polymer and break down the polymer, the mode of action usually we study that we can check and the polymer when it has microbial kind of activity, whether it has go and bind with the microbial protein, that kind of activity also could be studied. Am I clear?
Yeah, okay. I look into it, ma'am.
Yeah. Any other queries, participants? With this, the chemical structure preparation part, property analyzing part were completely over. From tomorrow, we are going for a target search. I mean, not tomorrow, Wednesday session, we are going to identify the target with the help of the chemical structure. Based on the session, we'll go with the topics. Any other doubts, participants? Any queries? No?
Dear participants, is there any questions? No.
Okay. Okay,
Could you
fine.
please give thumbs up then? I'm requesting everyone to give a thumbs up if you're clear
Yeah.
till now.
Okay,
Thank
fine.
you.
You just work
And
on and come back on Wednesday. We'll work with next databases. Thank you so much. And the
Dr.
Excel,
Shubha, I guess you will be uploading this PPT by today itself.
yeah, I'll just upload the PPT as well as the Excel file for the participants' reference. Sure. Thank you.
Okay, great. Yeah. So is there anything to add on?
They can come and prepare for the next class. Nothing else.
Okay, Doctor. Thank you so
Okay.
much. And thank you for joining today. Dear friends, please do fill out the feedback form and give your valuable suggestions about today's session. And as mentioned by Dr. Shobha, we will be meeting on Wednesday, 7:30 for our next session. So until then, thank you all. Please do prepare for what has been done so far. And kindly review the recordings as the recordings will be available only uh, uh, till the end. And you can just review it and prepare for your quiz as well. Thank you all once again. And thank you, Dr. Shobha.
Thank you, participants. Thank you, Bhavani.